Hi everyone. This is lecture 4 on neural integration, and to me this is probably one of the most challenging ones to describe because we kind of gave you the preview in the last lecture that unlike the neuromuscular junction, the synaptic transmission with neuronal cells are way more complicated because of the sheer number of synapses that are formed. Therefore, the way the result of the different synapses that form, which results in neural integration, is very complicated. So we're going to simplify it a little bit in this lecture, but try to give you a small taste of what's really happening in nervous tissue. Okay? Of course, we cannot do justice of the complexity of the brain and the spinal cord. Okay, so our major objectives for this lecture are to describe excitatory and inhibitory postsynaptic potentials. We introduced these terms last time, but I told you we will discuss them at a later lecture. And we'll give you some different ways that different pathways can form, such as convergence and divergence. Okay. We're going to see certain events such as temporal and spatial summation, which might look kind of familiar to you, and we'll describe a specialized system called a reverberating circuit. And the diagram, I think, in this figure is from a different textbook. Okay. And so that's the goal for this lecture. Okay. It's not a long lecture, but it's really a very important lecture for you to kind of understand the complexities that are involved in the nervous system. Okay, so let's begin with postsynaptic potentials. Okay, we know that a synapse is formed by a presynaptic neuron and a postsynaptic neuron. The presynaptic neuron, from an action potential, releases neurotransmitters. Okay, if it's a chemical synapse. As a result, the neurotransmitter binds to a receptor. Okay, which could be inotropic or metatropic. Okay, as we talked about last time. But as a result of it, a local potential is produced, just like you saw from the acetylcholine receptor, where a small amount of sodium entered this muscle cell to produce a small depolarization. Okay. But in nervous tissue, you can either have a small depolarization, or it can be opposite and produce a hyperpolarization. Okay. So as a result from these postsynaptic potentials, the membrane potential can move closer or further away from the threshold. Okay. So of course, it goes closer to the threshold, this can be excitatory. If it goes further away from threshold, then be inhibitory. Okay. So we have two different types of postsynaptic potentials. One can be excitatory, an EPSP, or excitatory postsynaptic potential, or maybe inhibitory, or IPSP, inhibitory postsynaptic potential. Okay? Let's look at excitatory postsynaptic potentials first, EPSPs. And these result from a small depolarization that moves the membrane closer to threshold. So we see here the resting membrane potential. Okay? And due to the neurotransmitter receptor, we get a small depolarization, and we see that it's getting closer to threshold, indicating it's an EPSP. Okay. Of course, if it doesn't reach threshold, then we know these local potentials will just reverse and go back to the resting membrane potential. Okay. But of course, if it reaches threshold, then this will open up voltage-gated channels to cause an action potential. Okay, so excitatory postsynaptic potentials, or EPSPs, are a result from openings of sodium and calcium channels, or calcium channels. I guess that would be a better term. Okay, which allow positive charges into the postsynaptic neuron. As positive charges enter the postsynaptic neuron, this is going to increase or make more positive the resting membrane potential. Okay. So in this diagram, here we see a ligand-gated sodium channel that, upon binding of the neurotransmitter, the channel opens to allow sodium in. On the right side here is a ligand calcium channel. And of course, once the neurotransmitter binds to it, it will allow calcium to enter. Since both are cations, the positive charge moving into the cell will raise the intracellular potential, making it more positive, thereby depolarizing the membrane. Okay. So now we can continue on with our synaptic transmission, because we kind of stopped here at number 3. So once the neurotransmitter binds to its receptor, it opens a channel, okay, which may be part of the receptor or separate. Here in this figure, it's inotropic because the channel is part of the receptor. It opens and leads to a change in the membrane potential from this local potential. Okay. Notice that it may possibly form an action potential. Okay, so these local potentials does not guarantee an action potential because it has to make sure it reaches threshold. Okay. So those are excitatory postsynaptic potentials. Let's look at inhibitory postsynaptic potentials, IPSP. Now these are result from small hyperpolarization of the membrane potential. Remember, hyperpolarization is becoming more negative, and if it becomes more negative, then we're getting further away from threshold. So it's going to be inhibitory. And moves the membrane potential of the postsynaptic neuron further away from threshold. So it's going down rather than up. Okay. Now, inhibitory postsynaptic potentials are generally produced by channels such as potassium or chloride channel. Because potassium moving out of the cell will make it more negative, okay, or chloride moving into the cell will make it inside more negative. So both of these will produce a inhibitory postsynaptic potential and a hyperpolarization, moving the membrane potential further away from threshold. Okay. So here is an example of a ligated potassium channel on the left and a ligated chloride channel on the right. So upon binding its neurotransmitter, movement of potassium out of the cell or chloride into the cell will both result in hyperpolarization and produce an IPSP. Okay, so make sure you understand why this positively charged potassium, as it leaves, is going to make it more negative on the inside. Okay, and these negative charged chloride ions, as they move in, is going to make it more negative inside to produce IPSPs. So let's look at a summary of synaptic transmission now. Okay. Remember, first we have an action potential. Okay. So we have an action potential coming down from a presynaptic neuron. Then we have synaptic transmission, because here, remember, there's calcium-dependent exocytosis of neurotransmitters that can be released into the synaptic cleft. Okay. As a result, they bind to receptors on the postsynaptic neuron and result in a postsynaptic potential, either an EPSP or an IPSP, depending on the type of neurotransmitter. Okay, so here on the left is a EPSP, on the right here is an IPSP. Number 4, we have termination of the synaptic transmission. We mentioned there were 3 different ways to terminate the transmission, you know, diffusion and absorption, reuptake, and the digestion of the actual neurotransmitter. Okay. So now we're all set up and we have all the tools and information to further describe neural integration. Okay? The neural integration is where the postsynaptic neuron evaluates all the information that it's receiving at the given moment, and it has to decide what to do. Okay, and the nervous system is so very malleable, or has many variables, that you can choose a number of different ways to respond to this information from the presynaptic neurons. Okay, so neural integration is a process of comparing all stimuli that affect a neuron that either excite or inhibit the firing of an action potential. So remember, in the neuromuscular junction, we only had that one nerve and that one muscle fiber. So it's pretty simple there. Well, what's the neuron to do if it's being bombarded with information of 1,000 different presynaptic neurons? And if they're all different, what should it decide to do? Well, that's what neural integration is. And that's why it's so complicated. So one possible outcome of neural integration is summation. Okay? And this may look a little familiar to you from muscle twitch. So let's see. Summation is adding the input from several postsynaptic potentials to affect the memory potential at the trigger zone. Okay? So summation links local potentials and action potentials. Okay? So here in this picture here, we have 1, 2, 3, 4 different presynaptic neurons providing information to this one postsynaptic neuron. Okay? And so the same thing occurs that we have discussed about. Action potentials reach the axon terminals of the presynaptic neurons. The released neurotransmitters open ligand-gated cation channels in the postsynaptic membrane. Okay? Once the ligand-gated cation allows cations to flow into the postsynaptic neuron, this results in an EPSP. Okay? The depolarizer of the EPSP spreads away from the point of stimulation because it's a local potential and towards axon hillock. So here's the axon hillock here. This is where the action potential, if it's produced, will start. Okay? If enough local potentials summate at the axon hillock to reach threshold, voltage-gated sodium channels open and an action potential results. So if each of these reach threshold, then there's no doubt that an action potential will occur. Okay. But what if they're sub-threshold? Okay, one by itself may not be enough to reach a threshold, but since we have 4 of them together, each of them may depolarize it sufficiently that when all of them together are depolarizing, then they could summate to reach threshold and we get an action potential. Okay, so here we have the EPSP summating. So by themselves, perhaps neither of them will be enough to reach threshold, but working as a team, all 4 together may summate to their EPSPs to reach threshold and we get an action potential. Okay, so this is summation. But there are 2 types of summation, temporal and spatial summation. Okay, let's look at temporal summation. Okay, this occurs when the neurotransmitter is released repeatedly from an axon terminal. Okay, so here we have a presynaptic neuron affecting a postsynaptic neuron, and here it releases neurotransmitter repeatedly. And so in temporal summation, since it's releasing neurotransmitters rapidly in succession, it does not have time to repolarize, and so it forms these stepwise increases until it reaches threshold. So this is an example of temporal summation, that the neurotransmitter is released at a high frequency over time in order to get to summation. Okay? So this results in a quick EPSP that propagates toward the trigger zone, and we get an action potential. But the importance here for temporal summation is that the EPSCs must occur in rapid succession for summation to occur. In other words, if the neurotransmitter is too slow in repeatedly being released, it has time, the postsynaptic neuron has a chance to go back, repolarize back to the resting membrane potential, and it will not reach summation. But since they're being released in rapid succession, in high frequency, they can be additive to reach summation, and we get an action potential. So this is temporal summation. So temporal time is important. Now how about spatial summation? This involves the simultaneous release of neurotransmitters from multiple presynaptic neurons. So this is sort of the way I described the initial figure. So here we have 3 presynaptic neurons. All 3 are insufficient to reach threshold, but together, the 3 presynaptic neurons together may be sufficient to reach threshold and then produce an action potential. Okay, so this is spatial summation. Here, it's through the physical orientation of 3 presynaptic neurons synapsing onto a postsynaptic neuron, and they all release their neurotransmitters at the same time. Neither one may be sufficient to reach threshold, but together they can produce enough EPSPs to reach threshold. Okay, so here are the 2 examples of summation side by side. Okay, here's temporal summation on the left, where the neurotransmitter is being released in rapid succession, and on the right is spatial summation, where you have 3 presynaptic neurons releasing neurotransmitters at the same time to produce individual EPSPs that may be able to reach summation threshold together. So make sure you could distinguish between the 2. Okay. Now, so IPSPs can behave just like EPSPs. So don't believe that temporal summation and that the both types of summation are only occur for EPSPs, but they can also apply to IPSPs together. So in other words, IPSPs and EPSPs can both summate, both temporally and spatially. Okay, so make sure you go back and understand those last few figures, because many students, have trouble grasping that concept. Now, to make things even more complicated, we have functional groups of neurons that work together as a network neuronal pool or a neural circuit. Okay, so they have a group of neurons that work together for a specific function, and these form networks called neuronal pools that perform a common function. Okay, and these neuronal pools are organized into functional groups called neural circuits. So here's an example of a neuronal pool. So here's a group of interneurons within the central nervous system. And so the information that's being processed by a pool depends on its synaptic connections. Okay, so here we have one here that's going to 2 postsynaptic neurons. Okay, here's this one going to 2 postsynaptic neurons. And in the middle here we have 3 going to 3 postsynaptic neurons. So here's fewer, but here there's more. So in other words, this one by itself may not be able to produce an action potential, but the one in the middle with 3 can possibly produce an action potential. The one on the left is too small too to produce an action potential, but these 2 outside ones together might be able to attain threshold and produce an action potential. So here's an example of a neuronal pool. Okay, so I kind of went ahead and described the figure already. So the center green ones have the highest number of synaptic contacts. Spatial summation is possible. and enough EPSPs are enough to trigger an action potential. Okay. The side ones, though, in yellow or pink, I guess it's yellow, but to me it looks pink on my computer here, have fewer synaptic connections. So alone they cannot produce an action potential, but only if both of them fire together. Okay. So neuronal circuits are patterns of synaptic connections between neuronal pools. How pools are connected depends on the function of the circuit. There are two basic types of neural circuits, two general types. I mean, there's many modifications of these. These are diverging and converging neural circuits. Okay. So in a diverging circuit, one axon of an input neuron branches with multiple postsynaptic neurons. So it's diverging means they're dividing into more contacts. When a signal is transmitted, an increasing number of neurons are excited. So let's take a look. So here's neurons in the brain signals to multiple muscle fibers, for example. Here they're diverging. Divergence because they're increasing in the number of contacts with the postsynaptic cells. Here they're muscle fibers. Okay. Likewise, an input neuron of the spinal cord sends the signals to different parts of the brain by the same process, but now it's going toward the central nervous system. And here this dividing is also divergence. Here it's sending information to different parts of the brain. Okay. Here, by divergence, it can produce a more powerful contraction because more muscle fibers are being affected. But in both cases, they are diverging circuits. So diverging circuits can be amplifying circuits, for example, in synapsing with different muscle fibers, or they can involve multiple tracks, like the second one where it was dividing and sending information to different parts of the brain. Okay. And so an amplifying circuit, the signal passes through a greater number of neurons. Okay. So here are these figures again. So this is an amplifying circuit because due to the divergence, we have 1, 2, 3, 4, 5, 6, 7, 8 muscle fibers that are now affected to produce a much stronger muscle contraction than if divergence didn't occur, you would only have one muscle fiber. Now in multiple tracks, these are more characteristics of sensory information from the spinal cord neurons to different neuronal pools in the brain. So here we have a sensory neuron sending information, but due to divergence, it can now send information to different parts of the brain. Okay? So for example, let me give you an example of this latter example. One of the most complicated senses is sense of olfaction or sense of smell, because so many effects can happen from the sense of smell. Suppose you smell cookies baking, okay? And in your brain, due to these diverging circuits, you're going to think about, oh, my grandmother used to make cookies just like that, and it's going to make you miss her and miss her memory. It might also tell you that you're hungry. Those smell of cookies are making you hungry. It might also tell you thinking about the recipe that's needed to make those cookies. Okay, so that's an example of different tracks of a diverging circuit. Now the opposite are converging circuits. Okay, so rather than spreading apart, they're coming together. So here we have axon terminals from multiple input neurons converging on a single postsynaptic neuron. Okay, so we start with 4, and then it goes down to 2, and then goes to 1, and this sends the information to a specific area of the brain. Okay, so multiple input neurons from sensory receptors converge onto a neuron in the brain. Okay, so an example might be having, let's say, many different touch receptors on your fingertips and allowing you to know exactly where the probe is that's stimulating them. Because You have so many receptors on your fingertips. So that's just one example. Now, finally, another type of circuit that's kind of specialized is called a reverberating circuit. And this is not in your textbook, okay? But I still think it's important. Reverberating circuits are neurons stimulate each other in a linear sequence. But one cell re-stimulates the first cell to start the process all over again. That's why it's called reverberating. Okay? So here's a reverberating circuit. So they're linear, like this, but one neuron branches off and re-stimulates one of the earlier neurons. Okay? So we can read this. Collateral branches of axons somewhere along the circuit extend back toward the source of an impulse and further stimulate the presynaptic neurons. Reverberation is like a positive feedback loop involving neurons. Once the reverberating circuit has been activated, it will continue to function until there's synaptic fatigue or an inhibitory stimuli break the cycle. So essentially, this linear sequence of neurons, a branch comes back to synapse against one of the early presynaptic neurons, and you get this endless cycle of positive feedback of going around and around in a circle until something stops it. Okay? So let me give you an example. Breathing is an example of a reverberating circuit because we want it to repeat. We don't want to just breathe once. We want to keep breathing to stay alive. So that makes sense to have breathing as an example of a reverberating circuit. Okay. Another example of a reverberating circuit is what's called the after image effects. Okay. So have you ever looked at a lamp with the light bulb on, and you stare at it for a while, and then you look away? You still see that glow, even though you're not looking at the lamp anymore. So that image is forming a reverberating circuit from your eye, and that reverberating circuit continues for a while as you look away. Okay, and so that after image is from a reverberating circuit. Okay, so this is the end of the lecture, but hopefully this is a small taste of how really complicated the nervous system is. Because we only showed examples of 3 presynaptic neurons affecting a postsynaptic neuron. But think about it when there's thousands. So that postsynaptic neuron is bombarded with all this information. That cell body has to decide what to do because all the information is differing. Some of it's antagonistic. And so it has to filter and decide what it wants to do. Okay? So try to keep this in mind and how complicated the nervous system truly is. And we try to give it in a kind of a simplified form here, but it truly is very complicated. And now we're going to set up to start looking at different parts of the brain and spinal cord functions. Okay, so this is the end of lecture 4.